Hello friends, welcome to Biology Extrema, where we teach biology from the basics to extreme. Myself Keshav Bansal, and today we are going to study about lipids, and specifically we are going to study about fatty acids. Okay, so before starting the topic, I wanna inform you that this channel Biology Extrema contains video in both English languages and Hindi language. Okay, so and most of the videos are dedicated to Hindi language. So we made a special playlist by the name of Life Sciences in English, where all videos are stored, which are in English language. Okay, topic. So what are fatty acids? First, we need to know that and how they are linked to lipids. Okay, so before studying fatty acids, we need to have at least some basic knowledge about lipids. So what are lipids? Lipids are heterogeneous. Lipids are heterogeneous compounds which are insoluble in polar solvent like water. Okay, polar solvent like water, and soluble in non-polar solvent like ether or chloroform. Okay, and why we call them heterogeneous group of compounds? Because like proteins, carbohydrates, and nucleic acid, they are not true polymers. They are polymers, but they are not true polymers. Because proteins, carbohydrates, these are made up of repeating units. Okay, like amino acids or saccharides. But lipids are heterogeneous group of compounds because heterogeneous means they are composed of different varieties of compounds, or we can say different varieties of components. Like if we talk about lipids, we can categorize lipids in two major parts. Okay, or we can say two major types. We can classify them as simple lipids, or we can classify them as complex lipids. Okay, so first talk about simple lipids. So in simple lipids are nothing. They are esters of fatty acid and alcohol. Means if we join fatty acid and alcohol, what will we get? We get lipid, and this is a ester because by joining a fatty acid and alcohol we get ester. So it's a ester of fatty acid and alcohol. This is called a lipid. So from this we know that there are two components, fatty acids and alcohol, and fatty acid is a component of lipid, or we can say simple lipid. Now if we add additional group in this simple lipid like proteins or carbohydrates or some phosphoric acid, so it will become complex lipids. So and there are different varieties of complex lipids. Okay, so now I think we have a good idea where fatty acids stand. It is a component of lipid. Okay, so now let's start. So in this video we are going to study what are fatty acids, okay, and their chemical structure, and what are saturated fatty acids and what are unsaturated fatty acids. Okay, it's the classification based on the structure. We'll talk about it. So we have already some idea that fatty acids are simplest form of lipids. Okay, and they are constituted in complex lipids. Clear? Now if we talk about the chemical structure of fatty acid, so we know that lipids are fatty acid plus alcohol if we talk about simple lipid. But what are fatty acid? Is their chemical structure? Okay, so if we talk about fatty acid has two components. It has a hydrocarbon chain. Wait a bit. It has a hydrocarbon chain. Okay, a hydrocarbon component which is attached to carboxylic group. Okay, carboxylic acid group. So hydrocarbon chain is attached to carboxylic acidic group. The hydrocarbon part is non-polar part. Okay, it's non-polar part means it's hydrophobic in nature, while the carboxylic part, it's polar part. It means it's water loving. It's hydrophilic in nature. So what we can see here that fatty acid. This is fatty acid. Okay, so fatty acid contains both polar, non-polar part and polar part. So a molecule which has both polar hydrophilic and non-polar hydrophobic part, we call it an amphipathic molecule. Okay, it's an amphipathic molecule. Okay, because it has both non-polar and polar domains. Now we know what polar are, what is polar domain and what is non-polar in fatty acid. Now we need to know that if we talk about this fatty acid, they can be branched. The hydrocarbon chain in fatty acids can be branched or it can be unbranched. Okay, and the number of, if you talk about the number of carbons in hydrocarbon chain, they can be even number or they can be odd number. Okay, even like 2, 4, which are divided by 2. I think you already know that. So if you talk about biological occurring fatty acids which are naturally found in living organism, which kind of fatty acids are they? So they are usually unbranched fatty acids which contain even number of carbons in hydrocarbon chain. Okay? So naturally occurring fatty acids contain even number of carbon atoms and their hydrocarbon chain is unbranched. Usually about they usually contain about 14 to 24 number of carbon atoms. Okay, now we have a good idea about what fatty acids are. Okay, their chemical structure. Now we need to understand what are saturated fatty acids and what are unsaturated fatty acids. Okay, so we already studied that it contains a hydrocarbon part. Wait a minute. It contains a hydrocarbon part. So let's draw a fatty acid. Okay, so this is the fatty acid. Okay, and this again is a fatty acid. Okay, so in this first case, if all the bonds which are present at also at the hydrogen, this is also CH2. Okay. CH3 we call methyl and CH2 we call methylene. Okay, methyl is this and methylene is CH2. So in the first case, all the bonds in the hydrocarbon chain, this is the hydrocarbon chain, all the bonds in the hydrocarbon chain are single bonds. Okay, so those fatty acids which have single bonds in the hydrocarbon chain, they are called saturated fatty acids. Okay, saturated fatty acids. Okay, now take a second case. Okay, now those fatty acids which have double bonds in the hydrocarbon chain. Okay, there can only be two possibilities, or either they can be single bond or there can be double bonds. There cannot be triple bonds. Okay, so if single bonds are present, they are called saturated fatty acids. If double bonds are present, they are called unsaturated fatty acids. What? Unsaturated fatty acids. Okay, now the number of double bonds can be one. There can be one double bond, so we will say monounsaturated fatty acid. Monounsaturated fatty acids. Okay, and if number of double bonds are more than one, okay, if they are more than one, then we call them polyunsaturated fatty acids. Okay, now there is no problem with monounsaturated fatty acid. We can the single bond the double bond can be anywhere in the hydrocarbon chain. But if we talk about polyunsaturated fatty acid, there need to be a special arrangement. Okay, what that is? Let's draw a hydrocarbon chain again. This is the carboxylic group. Okay, OH, and this one. Okay, so if we need to apply two double bonds, so what can be the possibility? Either they can be alternate single bond and double bond. Okay, like in this first case, or they can not be in alternation. Means a double bond means two double bonds are separated by a methylene group. Okay, like in this case, double bond, single bond, double bond, single bond. There is alternation between them. Okay, so they are called conjugated. They are in conjugation. So either this can be a possibility, or the double bonds have a methylene group, this CS2, this methylene group between them. Okay, these are two double bonds and there is methylene group between them. So which kinds of polyunsaturated fatty acids are possible? So answer is number 2. Okay, in the case number 1, there is conjugation, alternate double bonds and single bonds, which are not found in polyunsaturated fatty acids. Okay, so so case 2 is possible. So always remember, alternation, or we can say conjugation, is not possible. Okay, the double bonds will always, two double bonds will always be separated by a methylene group in polyunsaturated fatty acids. Okay, so this much is clear. And I think now we'll talk about some physical properties which are affected by the number of double bonds in the hydrocarbon chain length. Okay, so we take the physical property of melting points. We take physical property of melting points. Okay, so melting points is affected by two factors. If we increase the number of carbon atoms, if we increase the carbon atoms in the hydrocarbon chain, or we can say the chain length. Okay, we can say the chain length. If we increase the chain length, the melting point of hydrocarbons will increase. Okay, and if we increase the number of double bonds, if we increase the number of double bonds in the hydrocarbon chain, or we can say the degree of poly or the degree of unsaturation, then the melting point of fatty acid, polyunsaturated fatty acids will decrease. Okay, so this you need to remind that chain length is directly proportional while the melting point is inversely proportional to the number of double bond. Okay, so now we need to study that because we know that saturated, this is a complete lipid. Okay, in this picture this is a complete lipid, or it is a fat molecule, because fat is also a type of lipid. So we told you earlier that lipid contains a simple lipid. If we talk about them, these are fatty acid plus alcohol, and the alcohol can be anything. It can be a glycerol, or it can be sphygmocine alcohol. Okay, depends on the type of lipid. So this here in the picture we can see there is glycerol head. Okay, this is the glycerol head. So this is alcohol part. Okay, this hydrogen is alcohol part, and this is polar part. Okay, this is polar part. You need to remember that. Okay, now the fatty acid part is this. This is the fatty acid part. Okay, this is the fatty acid part, which consists of hydrocarbon chain, which is non-polar domain, and polar domain, which is a carboxylic group. Okay, which is, you know, that it is attached to glycerol head. Okay, so we talk about that saturated fatty acids contain only single bonds in the hydrocarbon chain. So there is no kink in them. What is kink? We will talk about it. In the saturated fatty acid, you need to understand that there is no double bond in the hydrocarbon chain, so there is no kink. Okay, while in the polyunsaturated fatty acids, or we can say the monounsaturated fatty acid, there is kink because there is double bond present in them. They can be a single kink if there is single double bond, but they can be multiple kinks, or if there are more double bonds. Clear? So what happens to the kink? You can see if we put these kind of structure. Wait a bit. If we put these kinds of structure, means saturated fatty acids side by side, okay, then these can pack very tightly. They can pack very easily. They will form a crystal lattice like structure. Okay, while if we try to pack unsaturated fatty acid, due to the kink, they cannot be packed tightly. Okay, there will be spaces. Will be loosely packed. They will not be tightly packed because there is kink, and due to kink we cannot pack them tightly. Okay, so this results in difference in their melting point. How? Because saturated fatty acids are tightly packed. So more energy is required to create a disorder in them. Okay, so due to that, melting point of saturated fatty acids are higher. Okay, due to that fact, melting point of saturated fatty acid is higher. While in case of unsaturation, in unsaturated fatty acids, due to because kink is present, they cannot be packed tightly, so they are loosely packed. So less energy is required to create a disorder in them. Okay, and that's why low melting point is there in unsaturated fatty acids. And now we also know that in the here we just learned that the number of double bonds, more the number of double bonds, more lower is going to be the melting point of fatty acid. Okay, so we now understand the concept behind it. Clear? Now we need to study about what are the examples. There are a lot of examples. We have just discussed some examples here, like palmitoic acid, oleic acid, arachidonic acid. These are types of unsaturated fatty acids. Okay, while if you talk about saturated fatty acids, they can be lauric acid, myristic acid, palmitic acid, and etc. Okay, there are more. Now there's also one point here which is mentioned that we know that fatty acids are attached to fat alcohol. Okay, fatty acids are attached to alcohol. They are esterified to alcohol in a lipid. Okay, but in vertebrates, free fatty acids can also be present. Okay, they can circulate in the blood, which are bound to serum albumin. Okay, but the majority of the majority of the fatty acids are esterified in the esterified form, or we can say are present in the lipids, esterified to alcohol. Okay, so this is some extra information you can remember. So our topic is complete. I hope you understand the topic, and in the next video we will discuss this, which is also written here, the cis isomerism. What is cis isomers? What are trans isomers? Because due to unsaturation, okay, the unsaturated fatty acids can be of two types. They can be cis fatty acid, or they can be trans fatty acids. Okay, so in the next video we will study about isomerism fatty acids. We will discuss the cis and trans isomers, and also study which type of isomers are present in biological lipids. Okay. I hope you enjoyed the video. If you enjoyed the video, please hit the like button, share the videos, subscribe the channel. You can support me in this noble work of providing free lectures and quality notes by donating only a small amount. Thank you and take care.